Hi, everyone. My name is Dwayne Mendes. I'm the CEO of DMark Biosciences, where for the past 18 years, we have been a Canadian-owned organization specializing in cutting-edge multi-omic solutions. I'm really excited by the seminar, and for many ways, this technology comes full circle for me and my research background. During my PhD, I focused on understanding a couple proteins using techniques such as in situ hybridization and immunohistochemistry in the brain, working to co-localize RNA and proteins. In my postdoc, I studied RNA binding motifs that regulate translation, really diving into the complexity of gene expression regulation. That experience taught me an important lesson. Just because an RNA is present doesn't mean the corresponding protein is being made. And even if the protein is present, it doesn't necessarily mean that it's active or that it's interacting with other proteins in meaningful ways. Studying function in a tube doesn't tell you anything about where those proteins are in or outside a cell. At DMark, we now offer technologies that help address those very gaps. For example, our Stomics technology, we can visualize thousands of RNA transcripts at the subcellular level, giving scientists unprecedented spatial resolution of transcriptomic data. But to truly understand cellular function, we also need to move beyond presence and quantity, and we need to move to protein activity. That's where Hawk Biosystems comes in. Hawk is a biotech company based in Bilbao, which is in the north of Spain, and originally spun out of the Francis Crick Institute in Cancer Research, UK. Hawk has developed QF-Pro, which is a powerful spatial bioimaging platform. What makes QF-Pro unique is that it doesn't just show where the protein is. It allows us to quantify protein functional states within fixed tissue samples. In other words, it enables us to see if proteins are actually doing their jobs, whether they're activated, phosphorylated, or engaging in critical interactions with other specific proteins. This fills a major gap in our ability to link molecular presence to functional relevance, something that's especially critical in cancer research, neuroscience, and complex tissue systems. At DMark, by combining technologies like Stomics and QF-Pro, we're moving closer to truly an integrated view of cell biology, from gene expression to protein localization to functional activity, all with spatial context. And so I'm very excited to first introduce Elena Castellano, who heads up research partnerships for Hawk, who will be the moderator of this webinar. Elena?
Thank you very much, Ian, for that kind introduction. Now I'm going to introduce James, who is our speaker of today. So James undertook his PhD between Hawk Biosystems, the University of the Basque Country, and the University of Bordeaux. During his doctoral studies, he used QF-Pro to assess the performance of PD-1 and PD-L1 interaction as a prognostic biomarker in a range of solid tumors. James graduated in 2022 with his PhD and continued his full-time role as product manager at Hawk Biosystems. Having worked with the QF-Pro technology since 2015, James has now helped with the design and launch of our QF-Pro products, including our reagent kits, our Violet 3.0 microscope, and our QF-Pro imaging software. So James, whenever you're ready, the floor is all yours.
Perfect. Thank you very much, Elena and Dwayne, and thank you everyone for attending. Yeah, I hope this will be a really informative webinar and seminar introducing Hawk Biosystems. I'll start just with a quick slide on who we are as Hawk Biosystems before moving on to our QF-Pro technology, how it works, a little bit of our products and how these help researchers to use QF-Pro. And then I'll move on to some cool research examples of where we've used QF-Pro and start to finish off with a really nice clinical example of what we've done with QF-Pro. And time permitting, I can also then present some results that benchmark our QF-Pro technology against the likes of proximity ligation assays. So, as Dwayne very kindly introduced, Hawk Biosystems, we're a biotechnology company spun out of the Francis Crick Institute and Cancer Research UK in London. And we've spun out into the Basque Country in Bilbao in the north of Spain. And what we do at Hawk Biosystems with our QF-Pro technology, and what we're very strong believers in, is that it's imperative to look at protein functional states in samples, whether that be cell samples or tissue samples. And that's because protein functional states are what drive disease manifestation, disease progression, and quite often protein functional states are what provide therapeutic druggable targets. It's very rarely just protein expression profiles that are responsible for this. So when we say protein functional states, we're spatially mapping and quantifying the likes of post-translational modifications, so phosphorylation states, methylation states, etc., or protein-protein interactions. And where we work with QF-Pro is, before I explain QF-Pro, I'll position where it is in relation to other technologies. QF-Pro works at a 1 to 10 nanometer distance, so it can almost be thought of as a biochemical ruler, which we use to quantify these protein functional states. This is unlike co-localization assays or proximity ligation assays that tend to work in excess of 40 and sometimes even 70 nanometers. So what is QF-Pro? QF-Pro stands for quantifying functions in proteins. And what we've done with QF-Pro is combine a two-site immunofluorescence labeling assay with forced resonance energy transfer, or FRET, and fluorescence lifetime imaging microscopy, or FLIM. And what we do is we label our two epitopes of interest. So let's imagine this protein-protein interaction. These would be labeled with two primary antibodies. The primary antibodies are in turn labeled with secondary Fab fragments. One is conjugated to a green fluorophore, which we term the donor, and the other to a red fluorophore, which we term the acceptor. Now, when these fluorophores are within 1 to 10 nanometers, with QF-Pro, we can detect and quantify an energy transfer from the green donor to the red acceptor. And therefore, we can quantify and we can map where our proteins are interacting. How this looks in images is if we take the example of this immune checkpoint, TIGIT and CD155, we can see in green, we're happily mapping the expression of our donor species, which in this case is TIGIT. We in red are mapping the expression of our acceptor, which is CD155. But here where we have the pink or the magenta pixels, this is where we're having that resonance energy transfer. So only in those magenta pixels or those pink pixels are we detecting a transfer of energy from our donor to our acceptor. And therefore we can confirm that in these areas, our proteins are indeed interacting. So what makes QF-Pro different? Because I'm sure a lot of you have probably heard of or even used forced resonance energy transfer and fluorescence lifetime imaging microscopy. Well, firstly, our assay is two-site, meaning we label two epitopes simultaneously within a sample, and we only detect and quantify areas where those two binding events have occurred. So this means the chance of false positives is greatly reduced, and we're less immune to the artifacts of non-specific antibody labeling events. Secondly, and what really pioneers QF-Pro is, unlike traditional FRET assays, we have performed a chemical amplification of our acceptor fluorophore. So this means that we drive the signal-to-noise ratio of QF-Pro up by about 4 times. Now, this is super important when using QF-Pro in tissue samples, because tissue samples, formalin-fixed paraffin-embedded samples from patients, as an example, have a very high noise, which is in part due to do with the fixative agents and a high autofluorescence. What this means is with our amplification of the acceptor fluorophore, that increase in signal-to-noise ratio means that for the first time, we have a reliable and robust FRET-based assay that now works very well in tissue samples. And critically, we've tried to make this as integratable into current laboratory and clinical workflows as possible. So the workflow can essentially be divided into 3 parts: staining, readout, and analysis. So what makes this really easy to use is the fact that, as I said, QF-Pro is based on immunofluorescence. So the staining, which can be performed with our reagent kits, which I'll explain later, this can be performed in FFPE tissue slices or in fixed cells in a 96-well plate, for example. So as I said, immunofluorescence styled labeling. These samples are then fed into a fluorescence lifetime imaging microscope, of which we have Violet 3.0. This FLIM device allows you to quantify and measure the FRET and where your proteins are interacting or are active. And then we have our QF-Pro software, where we can analyze those images, export the data, And we get two outputs, as you can see. We get a nice spatial image of where our proteins are and where they're functionally active. And also we get a quantitative QF-Pro score. So I'll get everyone to bear in mind the QF-Pro score, because when we see the research and particularly the clinical examples that I'll share later, this quantitative QF-Pro score becomes very important. So before I introduce the products and how users can access and work with QF-Pro, really, what can QF-Pro do? Instead of just saying protein functions, what kind of things do we look at? So as I've said already, the obvious are protein-protein interactions or post-translational modifications. We can look at protein DNA interactions, receptor dimerization, protein conformational changes, essentially anything where a user can probe two epitopes with two primary antibodies. In theory, a QF-Pro assay can be generated. And this has applications pretty much everywhere. So where a lot of our focus is in translational research or companion diagnostics, but this can, as you'll see, deliver into clinical diagnostics, or even it can go backwards towards more fundamental research to find out how signaling pathways are functionally active, how they're interacting, etc. So the Hawk Biosystems products, how does someone actually use QF-Pro? So the first product that we offer is the QF-Pro reagent kit. This contains everything needed to do the labeling of QF-Pro. Again, it's a two-site immunofluorescence assay. It contains secondary reagents, those critical amplification reagents for the FRET, blockers, quenchers, mounting media, etc. The only thing not included are the primary antibodies. So essentially, that means with any one QF-Pro reagent kit, multiple experiments can be performed against multiple targets. So we've tried to keep this as open as possible. Next, and where we really offer, we feel quite a strength, is in Violet 3.0. So as I said, in order to accurately quantify the threat that underpins QF-Pro, we need fluorescence lifetime imaging microscopy. Now, traditionally, fluorescence lifetime imaging microscopes have been pretty inaccessible for biologists and life science researchers. They typically need to be in a dark, air-conditioned room on a large, pneumatically leveled optical table. These softwares that are associated with them are not very intuitive or easy to use. And typically they need an experienced technician to run them for you. So really, that would put a barrier in the way of using QF-Pro and getting hold of those benefits. What we've done with Violet is try to break down all of these barriers. So we feel that we've probably designed one of the easiest to use FLIM devices out there. It's a benchtop fluorescence lifetime imaging microscope. It contains everything needed to do fluorescence lifetime imaging microscopy and FRET. So modulated laser, modulated detector. It comes with a range of dry objectives, so a nice 2x objective for overview scanning of samples, a 20 or a 40x objective for doing the QF-Pro acquisitions. And we've also combined a 4-channel LED system, so you don't just get a streamlined benchtop fluorescence lifetime imaging microscope, but you also get a routine immunofluorescence microscope as well. And we've optimized this. So essentially, all the user needs to do is turn it on at the side, open this lid, and insert either their microscopy slides or their multi-well plate, close the lid, and move straight onto the software. And again, as I said, typical or historic fluorescence lifetime imaging microscope softwares, and even, I regret to say, some of the modern ones, really aren't user-friendly at all. With QF-Pro, we've tried to just simplify this down into a workflow that a researcher of any background could work with. So this starts with logging details of the experiment, moving on to the acquisition tab, mapping areas of interest within a sample, pressing go, and then moving on to the analysis and the export. So upon pressing go, the system will automatically take all of the images and extract all of the data from the sample. The images can then be analyzed if one wanted to look and adjust some thresholding or some brightness and contrast, etc. And essentially you can export those spatial images that I said, and also an Excel table of all the quantified functional proteomic data as well. So we really feel that we try to get this, as I said, as integratable as possible and really remove the barriers that FLIM has typically put in fundamental and translational research applications. But enough of the theory on what we've done with QF-Pro and how it works, but really, what are the applications? What examples do we get in the real world of experimenting with QF-Pro? So in this first, I'll run through, I think, 3 examples we have here. In this first example, we're looking at a protein-protein interaction in 2 patients or 2 triple-negative breast cancer patient samples. So what we've looked at here is the interaction state of E-cadherin and beta-catenin. And what we can see is in both patients, in the immunofluorescence images, we have a good expression of beta-catenin and a good expression of E-cadherin. However, only in patient number 2 do we spatially map these red pixels, which is the interaction. So only where these red pixels are do we have a resonance energy transfer, i.e. QF-Pro is working, and only in those red pixels are beta-catenin and E-cadherin actually interacting. And so, as I said, this is nice and easy to map, but really where our key strength is being able to quantify that data. So we build up a really nice statistical plot showing the significant difference in interaction state between patient 1 and patient 2. Critically, if I was to just take these 2 images of patient 1, merge them and look at them in ImageJ, for example, in a more co-localization manner, I would probably infer that there was interaction happening everywhere, sorry. But if I just did that and co-localization is only working at 70 to 100 nanometers, well, that's giving me a false positive result. And it means that I'm not really seeing a true interaction if I just do a merge of these 2 images. So it's really only with that 1 to 10 nanometer resolution of QF-Pro that we're accurately picking up an interaction state of these 2 proteins. Another example, and we've gone back a little bit more fundamental here towards cell models. We've looked at that immune checkpoint TIGIT and CD155 in a cell co-culture model. And again, we can see here red pixels showing an interaction state of our 2 proteins. But upon applying or treating the cells with a blocking antibody, we remove these red pixels. So again, we can map that we were able to modulate that signaling pathway and disrupt that interaction state. But again, critically, with that quantification, we can quantify the reduction in signal that we observe. And critically, we think that being able to see these 2 points here, that really could give some insight as to where that experimental drug maybe isn't penetrating, where maybe the pharmacokinetics or dynamics are slightly different. So really, this quantification is the real advantage that we feel that we bring with QF-Pro. And I think the last research example that I have is, rather than a protein-protein interaction, is a protein post-translational modification. This was looking at the phosphorylation state of AKT, or also known as protein kinase B. And here what we did is label all protein kinase B, and then with our acceptor, we labeled the phosphorylated threonine 308 residue, which is what is phosphorylated when protein kinase B becomes active. And we did this in SKBR3 breast cancer cells, and we did a time course stimulation of these cells with epidermal growth factor for 0, 1, 2, 5, and 10 minutes. So what we can see again with these images is it's very easy to see that color shift in this scale from blue down to green and yellow. So very visually, one can see that the EGF has increased the activation state of protein kinase B. The orange pixels that we have around the plasma membrane, these are showing the areas of the highest activation state. So we can map that actually protein kinase B is undergoing its activation kinetics predominantly at the plasma membrane. But again, with that quantification, we can build up a really nice dose response, or I should say time response curve, of how protein kinase B activation is increasing in the presence of epidermal growth factor. So again, as we'll see in some slides later, other technologies that lack this quantification are unable to build up these kinds of results. So that's a couple of research examples of what we've done with QF-Pro. But now I'll just talk to you about how we try and extract as much data from a given QF-Pro experiment as possible. So if we look at the protein interaction of signal A and signal B here, of course, we get the interactive data of protein A and protein B. But we also can report on the expression of signal A, the expression of signal B, and we could add a third biomarker for expression profiling of signal C. So if we look at the immune checkpoint TIGIT and CD155, we could map only in areas where we have CD8 positive cytotoxic T cells, for example. So we can mix and match a little bit of what expression data we get from a QF-Pro assay. And of course, we can also add DAPI nuclear staining. Critically, we want to be able to extract as much functional data from a sample as possible. So we have engineered multiplexing into QF-Pro. So after doing this labeling and analyzing this data, it's possible for us to remove these signals and then reprobe against a new set of targets. So we can build up another panel of expression and functional data. And at the moment, in a given slide, we can build an image of up to 4 functional states and 13 expression profiles in a single sample. So how this may look, as an example, if we look in the domain of immuno-oncology, we could see in this given patient tissue sample that we have lower interaction state of PD1 and PD-L1, a higher interaction state of CTLA-4, CD80, and again, the interaction states of LAG3 and MHC class 2. So again, we get the nice spatial images of where our functional events are occurring, and indeed our expression profiles are occurring, but we can build up that quantitative data. And again, later in the presentation, I'll show where that quantitative data becomes really critical. So just to give a flavor and kind of throw some names out there of the kind of biomarkers we've worked with QF-Pro, as you're starting to probably imagine, we have quite a good data set on immune checkpoints. So we've looked at PD1, PD-L1, LAG3, CTLA-4, and TIGIT. We've looked at several post-translational modifications, so not only the phosphorylation state of protein kinase B, but also STAT3 and Wnt. We've looked at dimerization events, intracellular protein-protein interactions, and many other biomarkers that we're unable to disclose. So really, this is just to reaffirm the flexibility of QF-Pro and help just to give a flavor of what we can do with it. But again, to repeat, anything in theory where we can target 2 epitopes with 2 primary antibodies, a QF-Pro assay should be able to be created fairly easily. And if someone follows the QF-Pro workflow all the way through these translational research applications, where can it bring research? So we've got a clinical case study of what we've done with QF-Pro. And I think this really helps to highlight that if researchers take the correct quantitative proteomic approach to their research and to their samples, this is the kind of data that can be generated. So, as I said, we undertook a clinical study, and this was done in the domain of immuno-oncology. And we wanted to help hit an unmet clinical need, which is the low response rates that are observed to immune checkpoint inhibitors, particularly drugs targeting the PD1, PD-L1 immune checkpoint, and the low response rates that they exhibit in solid tumors. This depends on the solid tumor, but the response rates can be as low as 12 to 25%. So really, these are really powerful drugs that can help reactivate the immune system and allow the immune system to detect and target and clear cancer cells or tumor cells, but the response rates are really low. And it's thought that one of the drivers behind this is an inability to correctly stratify patients to receive these immunotherapies. So we looked in non-small cell lung cancer, and this was the study we presented in the Journal of Clinical Oncology in 2023 in 188 NSCLC patients. And what we hypothesized is the current gold standard biomarker, which is an immunohistochemistry score that looks at the expression of the ligand PD-L1. Essentially, if you have a high expression of PD-L1, you'll be put forward for a drug to disrupt PD-1 and PD-L1 signaling. if you have a low expression of PD-L1, you wouldn't be given that drug, not at least first or second line. What we hypothesized with QF-Pro is that really looking at the expression of PD-L1 isn't really biologically very relevant. And if we use QF-Pro to measure the interaction state of PD-1 and PD-L1 before the patients are given an anti-PD-1, PD-L1 therapy, well, surely that would be much more predictive of whether they will respond or not. And essentially, that's exactly what we saw. So in this study of 188 patients, they all had biopsies taken at the time of diagnosis or surgical resection. And we received these samples before any anti-PD-1 and PD-L1 therapy was given. So we could then track these patients' overall survival and or progression-free survival to see how they were responding to the then prescribed anti-PD-1, PD-L1 therapies. What we first saw is what has been confirmed by numerous other labs and publications, that the PD-L1 TPS, tumor proportion score, that immunohistochemistry test of PD-L1 expression, really isn't very predictive of response to an anti-PD-1, PD-L1 therapy. Sure, we have a trend that higher PD-L1 expression has a slightly elevated, those patients have a slightly elevated response to these therapies, but this is a trend at best and is completely non-significant. And we really feel that this is one of the main reasons that response rates for these therapies remain so low. However, when we quantify the interaction state of PD-1 and PD-L1, which is the very target of the drugs, when this was quantified using QF-Pro, we showed that the patients who had the highest interaction state of PD-1 and PD-L1 had a significantly better response to therapy with a median survival of 31 months compared to the patients who had a low PD-1, PD-L1 interaction state, who had a severely reduced median overall survival of just 10 months. So we showed that with our technology and with this approach, we're able to significantly predict the response to anti-PD-1 and PD-L1 therapies. And critically, that quantification was really important for us. So not only could we just separate those patients out, but we could also give a clinical cutoff threshold value of 2.13% of where patients above this would significantly respond more to these therapies compared to the patients below. And again, in data that I'll show later, technologies that lack this quantitative approach are unable to provide these kinds of clinical results. And in the exact same cohort of patients, but just plotting them based on their interaction state or their expression state of PD-L1, we could actually see that 24.5% nearly of patients had a low PD-L1 expression score of less than 50%. But despite this, they had a high PD-1 and PD-L1 interaction state. So this shows that nearly 25% of patients under some health guidelines would not be prescribed these therapies first line. However, they would be very, very good responders. So this is 25% of patients almost who are missing key therapeutic windows of opportunity to receive drugs to which they would respond. But on the other side of the coin, this 22.48% of patients who have a high expression of PD-L1, so above 50%, these patients are given these therapies routinely first line, but despite having that high expression, the PD-L1 isn't necessarily interacting significantly with PD-1. So these patients actually still had a low interaction state, and therefore the drug had nothing to act on. And these patients are given these therapies for no therapeutic benefit, but are still exposed to those potential adverse effects. And really interestingly, because we know that the threshold to prescribe treatment of greater or less than 50% PD-L1 score may have changed in some healthcare systems, and people are starting to move towards going above or below 1% of PD-L1 expressing cells. Well, even in the PD-L1 less than 1% group, so these patients have really no PD-L1 present from an immunohistochemistry point of view. Despite this, at that 1 to 10 nanometer spectroscopic level of QF-Pro, we can still identify a good amount of responders who, again, under these guidelines, would still be missing key windows of therapeutic intervention to which they would respond. So that was, as I said, published in 2023. Now, today in 2025, we have increased that data set to, I think actually above now 240 patients, and the trends are staying the same. So we have the same cutoff value, the same median overall survival, and the same significant differences. So this is really just getting stronger and stronger with the more patients that we're adding. And what we're looking to do now is to continue to increase that N number and try and get this PD-1, PD-L1 QF-Pro assay IVDR certified, so we can get this deployed into clinics where it really can make a difference and start to help patients. And the kind of numbers that this could potentially achieve, we ran some calculations, and if QF-Pro was the first line stratifying test for patients to receive anti-PD-1 and PD-L1 therapies, we could potentially triple the efficacy and the response rates to these drugs, such as pembrolizumab and nivolumab. And critically, we could potentially double the survival rate of non-small cell lung carcinomas. So I really hope this shows that if QF-Pro and this kind of way of working and looking and quantifying protein functional states is adopted, that really the benefits could be huge. And again, where does that fit into our approach to try and look at as much data as possible? Well, it's not just PD-1 and PD-L1 that we're interested in from an immuno-oncology point of view. We could also look at TIGIT CD155, CTLA-4 CD80, LAG3 MHC class 2. So really, we're starting now to move to not just looking at PD-1 and PD-L1, although it's very important, but using QF-Pro to unveil and generate a functional immune signature on a per patient basis, which we think would be a real step towards precision medicine for immuno-oncology. And how that may look with some real world data that we have submitted and we're trying to get published is we looked in a cohort of 67 non-small cell lung cancer patients who have been treated with PD-1 and PD-L1 therapies. And we, in these patients, quantified the interaction state of PD-1 and PD-L1 and the interaction state of CTLA-4 and CD80. And what we could see was that the patients that were had high interaction states of both PD-1 and PD-L1 and CTLA-4 CD80 had a much, much better response to anti-PD-1, PD-L1 therapies than those patients who were low for both checkpoints. So whilst these patients were not treated with an anti-CTLA-4 CD80 therapy, we could nonetheless identify populations of patients who would respond very, very strongly to dual immune checkpoint inhibitor therapy targeting both PD-1 and PD-L1 and CTLA-4 and CD80. And we really think that the more patients and the more checkpoints we start to investigate, the more we can build up real world data like this, where we could identify patients who would respond to these dual or combinational immune checkpoint inhibitor therapies. And yeah, that's essentially where we want to go. So to really start looking clinically now at TIGIT, CD155, TIM3, GAL9, etc., and not just to look at non-small cell lung cancer, although it has a huge unmet clinical need, but move on to the many other solid tumors where immune checkpoint response rates are low. So breast cancer, melanoma, head and neck squamous cell carcinoma. Essentially, we're now looking to broaden this as much as we can. And essentially, that's it for the first half of this webinar, in the sense that I hope that I've nicely introduced QF-Pro, the products, the research examples, and really where that research approach can take us, which is to this really cool and powerful clinical data. But I will now move on and just show in a few slides how QF-Pro compares to other technologies, such as proximity ligation assays, which also aim to look at protein functional states, but they work in a very different way to QF-Pro. So as I said at the beginning, QF-Pro has an operational range of 1 to 10 nanometers, and that is the constraint of the forced resonance energy transfer. But proximity ligation assays, which also look at two targets, That does a rolling circle amplification between hybridizing oligonucleotides. And in theory, that will give you a nice red dot in an image wherever you have an interaction state. But that lacks quantification and has an operational range of often exceeding 40 nanometers. So we wanted to do a benchmarking series of experiments where we compared both technologies in the same samples to see how they compared. So we started with a first phase to do a technical benchmarking in a cell model, which was the SKBR3 breast cancer cells with protein kinase B activation state. We then wanted to look at the performance of both technologies for looking at a protein-protein interaction in a cell co-culture model. Again, something that we could modulate with a drug. And then a third phase to look at the performance and clinical utility of these two technologies in patient tissue samples. And what we can see is you've already seen these cell images in a previous slide, but when looking at the activation state of protein kinase B in these SKBR3 breast cancer cells, again, that change from blue to green and yellow was very clear to see that, okay, upon stimulating these cells for 10 minutes with epidermal growth factor, it's very easy to see that an effect has taken place and to map that the highest activation was in those orange pixels at the plasma membrane. With the PLA, however, what a user is recommended to do is look how many red dots there are per cell. And in our hands, when we ran this experiment, it really wasn't very easy to really distinguish which had the highest number of red dots per cell. In our hands, it actually looked almost like the basal cells may have had more dots per cell than the stimulated cells. Nonetheless, we were recommended to do a quantification of this proximity ligation assay result by using ImageJ to analyze how many dots there were per cell. And again, I'll start with the QF-Pro. When we stimulated those SKBR3 breast cancer cells with epidermal growth factor, our quantitative approach and our 1 to 10 nanometer operational range allowed us to build up this really nice time response curve. However, in this example, when counting the number of dots per cell, we couldn't see any significant differences between the groups. So really, it was super hard, very labor intensive to be able to even attempt this quantification. And when we did, it really didn't give very convincing results. In the second phase, where we looked at the interaction state of PD-1 and PD-L1 in a cell co-culture model in the absence and presence of a blocking antibody, well, again, with QF-Pro, we could very clearly see the abolishment of that interaction state upon treating the cells with this anti-PD-1 and PD-L1 therapy. And again, with the proximity ligation assay, we were unable to get any significant differences. I mean, as you can see, the error bars did reduce very nicely between the three groups, but still we didn't get any significant differences. And again, we think it's down to the distances at which these assays operate and the approach to quantification. And in this third phase, where we've looked at PD-1 and PD-L1 again, but in non-small cell lung cancer, again, the shift from blue to green is very visual and very easy with QF-Pro to see how PD-1 and PD-L1 interaction states have changed between these two different patients, and also to map in this patient these hotspots of PD-1 and PD-L1 interaction states. And again, with the PLA, it really isn't very easy by eye to see which condition necessarily has the most or least PD-1 and PD-L1 interaction, as determined by those red dots. For example, you see the red dots here, depending on the screen in front of you. It's really not easy to see if that's three separate interactions or one big interaction. Likewise, you have a bit of a shadow of red down here. It's hard to know, is that a real interaction? Is that non-specific, some non-specific binding? It's really hard to tell what that is. And what we actually did, the next step of this was to take 19 patients from our previous study, where we had some spare tissue samples from the non-small cell lung cancer cohort. And I think that was N equals 19. And just, again, run the PLA and see how that compared to QF-Pro. Now, I'd give The biggest disclaimer that, of course, this is n equals 19, and I don't think any conclusions would ever be drawn from this. But it was very interesting to see that QF-Pro still actually just hit significant differences and could still predict the patients with a high interaction state had a better response to PD-1, PD-L1 therapies compared to patients with a low interaction state. Again, n equals 19, so it's not to say that the addition or removal of a couple of patients would change that significance, but still I think that shows how robust this technology is. And the proximity ligation assay, we're afraid to say, absolutely was unable to yield any trends or any significant differences in response rates between groups. So really, as I've said, it's the sensitivity and dynamic range of QF-Pro that allows it, we feel, to generate these kinds of quantitative functional proteomic data sets. It's really very well advanced in terms of clinical validation, as you've seen with those results that I shared earlier. And unlike proximity ligation assays that need lots of enzyme cycling and lots of enzyme-based reactions, our technology being just essentially immunofluorescence based, we really feel helps it to be quite easy to use and fairly easy to integrate, as I said, into current research and clinical workflows. So yeah, I think with that, I think I'm a little bit ahead of schedule actually, but with that, I'd like to thank everyone for their attention and hopefully take some questions and discuss further.
So thank you very much, James, for this nice presentation. We actually have a few questions for you that I wanted you to answer on live. So the first one is from Kelly. He says, very impressed with the technology and its potential. Quick question. Do you have experience with trying to detect interactions between epitopes on the extracellular phase of transmembrane proteins and signaling epitopes of the cytoplasmic phase of the plasma membrane? Or is it the distance too far or otherwise impeded? Also, he then added another comment, maybe to clarify, since plasma membrane are about 10 nanometers, I'm wondering whether the length of the antibody layers may give you a bit of a spacer to detect somewhat greater distances than 10 nanometers between actual epitopes.
That's a fantastic question. It's a detailed question. That's a good one. I think so. My internet cut slightly as you asked it, but I think the crux of the question was, could we operate across the plasma membrane with QF-Pro essentially? That's a great question. My gut feeling would be probably not. I think the distance and the size of the plasma membrane itself probably is going to be disruptive to QF-Pro. When we look at intracellular interactions, or activation states such as protein kinase B and AKT activation states, or STAT3 dimerization, whatever it may be, we do a permeabilization of the plasma membrane. But if you were looking at things that if you're looking at epitopes you would expect to be localized towards the plasma membrane, such as when we do PD-1 and PD-L1 interactions, you wouldn't really want to disrupt the plasma membrane because you want to actually spatially see where things should be. So yeah, my gut feeling would be that probably the plasma membrane would be disruptive, but nevertheless, that'd be something I'd be actually incredibly excited to try and see how it worked.
So one of our next question is from James. He's asking, what is the negative control for the Fred film experiments? So for example, if you want to measure Fred between two proteins localized to the plasma membrane or another organelle, you do expect to measure some amount of Fred. So how do you know your Fred signal actually indicates a protein interaction and not just collision? Is that where the blocking antibody is for?
Yeah, that's it. So wherever possible, if you can modulate your signaling pathway of choice, and I'd probably, let me scale that back. When starting a new QF-Pro assay, we would always start it in cells where possible because we can modulate those signaling pathways a lot of the time. So yeah, completely, if you're able to target your protein of interest, or say the interaction state of protein A and B, if you have a nice way of being able to block and disrupt that interaction biologically, that becomes a great negative control. Other controls that we can do with the frets and the FLIM is simply to emit one of the fluorophores, so emit the donor fluorol or emit the acceptor fluorophore, and then you should be able to see that the fret is also reduced. So if you were to remove the labeling for protein B and just look at protein A, you'd see its expression and you'd see the donor lifetime of protein A everywhere and of that donor fluorophore, but you wouldn't see it reduce. So you do a mix of those secondary controls combined with, where possible, some biological manipulation of said signaling pathway.
Thank you very much for that detailed answer. So one of our next questions that we had in the chat box is, how does QF-Pro compare to phosphoprotein detection methods or single-cell proteomics when it comes to capturing pathway activation and signaling dynamics?
Falls slightly outside of my expertise, but when we've looked at protein phosphorylation before, this was actually the reason that QF-Pro was first developed is that our founders and the researchers who developed the technology wanted to look at the phosphorylation state actually of protein kinase B, and because they really thought that the phosphorylation state of that kinase would be prognostic in breast cancer. And the only tools that they had available was a one-site immunofluorescence assay where the phosphorylated residue was targeted with a primary antibody, with a fluorophore, routine immunofluorescence. And they saw that it correlated with absolutely nothing, that they were essentially seeing that fluorophore everywhere. And that's because of the drawbacks of just using a one-site assay, which was that antibody against phospho PKB was hitting PKA and PKC as well. So really, that's how it compared to the traditional way of looking at a phosphorylated residue, which would just be standard immunofluorescence or immunohistochemistry. As I said, our two-site approach, yeah, there may be some non-specific labeling or binding of that antibody, but we're only going to detect the threat where both our antibodies are on the same molecule there. So I think to the best of my expertise, I hope that answered the question.
So thank you very much. Moving on to another comparative question is the following. Can QF-Pro identify functionally distinct cell populations that may appear phenotypically similar in conventional multiplex immunohistochemistry or single-cell RNA sequencing?
That's a good question. I think it should work well. We've not combined QF-Pro with single-cell RNA sequencing. So it's something we'd have to try, but I think, yeah, we should be able to detect, I would hope that we'd be able to, with QF-Pro, detect those proteomic differences in those kinds of cells populations. So yeah, I'd be quite confident that should work nicely.
Mm-hmm. And then another one, which is more technical into the QF-Pro. So how is the QF-Pro score calculated?
That's a great question. Essentially, it's just a threat efficiency, or what's known as a threat efficiency calculation. So what we do is we take the lifetime of the donor on its own. Then we take the lifetime of the donor when it's in the presence of the acceptor. And we just run a small calculation that looks at those differences and turns it into a percentage. So essentially, it's the percentage of how much of the potential energy that can be transferred from the donor to the acceptor is actually transferred.
And our final question that we have in the box is, what is the difference between using PLA and QF-Pro to study protein-protein interactions? That I guess you have explained a little bit into the last part of the presentation, but if you want to go a little bit more into detail.
Yeah, of course. The main difference is twofold. It's what I said. It really is just the distances at which they operate. If QF-Pro can tell you whether two people are in a room holding hands, PLA will tell you whether maybe you have two people in neighboring rooms at best. So it's kind of just the distances they're working at.
James, I think we cannot hear you anymore. I cannot. I don't know if...
Can you hear me now?
Yeah, no.
Okay, perfect. I nearly got the whole way without the headphones breaking. So essentially, yeah, it's those working distances of QF-Pro is two people holding hands in a room as an interaction. PLA are two people in separate rooms at best in a building, coupled with the that quantification, which is the fluorescence lifetime imaging nature of the QF-Pro assay makes it spectroscopic essentially. Whereas with the PLA, it's you can automate it, but it's essentially dot counting. So yeah, they're the real differences that underpin the technology.
Thank you very much. Oh, we have a couple more questions coming right now. So the following one is HOC aiming for the regulatory approval within the PD-1, PD-L1 clinical study that you have shown?
Yeah, absolutely. Absolutely, yes. So I think that slide may have been slightly out of date. I think we're now up to about, we published the data of 188 patients, which was essentially the learning cohort. We've taken that up now to, I showed 240 patients, but I think we're now on 250 patients. I think we're aiming to take that somewhere between 600 to 800 patients of non-small cell lung cancer. And yeah, very much so would be to get that IVDR cleared and clinically approved for diagnostic use.
And is that going to happen both in Europe and the USA?
Yeah, I believe the pathway we'll take would be Europe first, then the USA. But yeah, very much we're aiming to get this into all major healthcare systems as quickly as possible. Essentially, when we run those numbers of how this could help patients, is really a case of getting it out there as quick as possible. So yeah, completely in the FDA approved as well.
Then another question that we receive is, does the QF-Pro software analyze and export data automatically in different formats?
Yep, that's a great question. Yeah, it does. So it will export the images as just a routine PNG file, so you can copy something into a PowerPoint presentation for a lab meeting in seconds. Also into TIFF files if anyone wanted to use other applications to look at the images. And that quantitative data is exported directly as an Excel sheet. So yeah, that's all seamless. It's just literally a click of a button and everything is batch exported. We actually, a couple of weeks ago, used another FLIM device, which was incredibly powerful, but lacked a button to export any data into Excel. So yeah, the QF-Pro software has that covered.
And one of our latest question is, does that mean the higher the QF-Pro score, the better the proteins are interacting?
Oh, great question. Yeah, sorry. Absolutely. Yeah. So the higher the score, the more interaction state is occurring. Yes. So higher the score, the more energy is transferred between the donor and the acceptor across a sample. So yeah.
And actually, we had a question before that I answered in the chat, but if you want to explain a little bit more, it's about the acquisition time of the QF-Pro and the Violet. So how long do we normally take to acquire the samples?
My standard response is always depends how big the sample is, but the detailed response is one image would take 16 seconds. So for a typical FFPE tissue slide, we're normally looking at 15 to 30 images to cover a representative area of that tissue. So I can never run that maths quickly enough in my head, but that would be looking at about 6 minutes per sample for FLIM image analysis. So for a fluorescence lifetime imaging microscope, it's incredibly quick.
Thank you very much. So I think with that, I don't know if we have missed. No, I think we have gone through all the questions now.
Actually, James, I do have one more question. It was just about the Violet 3.0. Can you do standard immunofluorescence experiments using that instrument?
Yeah, absolutely. That's a great question. So yeah, that we included a 4-channel LED system as well and all the corresponding filters. So that can just do routine immunofluorescence on top of the FRET data as well.
Okay, excellent. If we don't have any more questions, I think we could finish up early. I want to thank our speaker, James Miles, for highlighting the importance of this exciting technology, and of course to our moderator, Elena Castellano, for facilitating and providing answers in the background. As a reminder to all attendees, we at DMark have our JEXI Symposium at the University of British Columbia on May 8th. We have a number of different sponsors, including PacBio, Element, Covaris, QuantumSci, all exhibiting at that symposia. We look forward to seeing you there. We'll have another one at McMaster University in Ontario later on in the year as well. A link to the survey for this webinar will be sent out to all of you who registered, and we'll also upload a copy of this webinar onto the DMark website. So again, we want to thank you for all attending and look forward to hosting another webinar in the near future. Thank you again, James and Elena.
Thank you everyone
very
for attending.
much.
Thank you for the opportunity. It's been great.